Alright guys, welcome back to another tutorial. This one's going to be covering this is going to be covering regulating the cell cycle. Alright, this is your last tutorial of Unit 3. And it's going to cover a lot of the same things we've already covered, so I don't think it'll take very long. I'm just going to go into a little bit more detail about like S phase and M phase and all those different things. So if we start with page 5, why cells divide, the first reason is there's too much cell and not enough protein or not enough DNA. So one of the main reasons that cells have to split is because, through that synthesis phase, they're going to grow, starts like this, and they're going to get a little bigger, and then they're going to get bigger. But the point is they keep getting bigger and bigger, and they have too much stuff in them. And you have, your nucleus stays the same. So here's your nucleus, here's your nucleus, here's your nucleus. I guess it's normally drawn a little bigger than that. There you go. So your nucleus is going to stay the same size. Your brain can't control all that all that body, so you have to split it apart into two different cells eventually. Okay, so you have to synthesize the DNA, and then you have to reproduce. That way, it has a consistent amount of brain to cell ratio, essentially. Next thing is you have to have. You cannot take in that much food. Like, that's just how it happens. So, like, you might have the same amount of food all the time because, it's not like your cell membrane is changing the way it works. It still takes in the same amount of food, right? Well, if you take in the same amount of food, you'll notice that the bigger the cell gets, the less and less you have. So your second problem is not enough food. Not enough materials to sustain a big cell. You can think of a cell kind of like, kind of like a job. Point is, you don't have all that money in a job. Like, even if you're the boss or whatever, you just don't have the budget to keep everybody. So this is kind of like layoffs. So essentially, you're laying off, and then this company has to split into a second company. This is actually better as a stock example, but I'm not going to go into all that because this isn't an economics class, right? So you'll notice that page 6 is going to cover what happens exactly, and we've already covered interphase, mitosis, and cytokinesis, so I'm not going to worry about that. But on page 6 there is a wonderful picture that you can take a look at to visually see how everything changes. We actually are going to draw this out in the next tutorial and whatnot, and we definitely talked about it verbally in the last tutorial, so I wouldn't stress it too badly. Let's see. Page 7 just explains a couple of things like how injuries heal and why it's important to control cell growth, but I'm not going to worry too much about that. Feel free to read over it if you like. Page 8 is a question. Page 9. Okay, cool. Here we go. The first thing we're going to introduce are cyclins. And cyclins are proteins, and they're just going... Excuse me. They are going to cover the timing of the cell cycle. Basically, cyclins, you can think of like... let's see, where's the pen? Let's go blue. Cyclins are responsible for cyclins, like that, are responsible for timing. You get this word from the word cycle, and you guys know that cycles happen, in patterns and whatnot. So cyclins mess with that timing. Okay, so they do this with these checkpoints, and these checkpoints are normally during interphase and whatnot, and it's that whole G2, G1, S2, S1, that kind of thing, interphase, mitosis, whatever. So let's talk about what they are. They are timing proteins, by the way. Then there are types of regulators. So on page 10, you'll see two types of regulators. There are internal and external regulators. Go ahead and write this in orange. So internal regulators check what's going on inside a cell. Make sure the inside's cool, because internal means inside, right? So make sure the inside of the cell is ready for the next step. Inside of cell is ready, and this is for the next step and all that jazz. On the other hand, external, so we're going to go back to blue right here. Well, actually that's the same as cyclins. Let's go ahead and go green, I guess. External, I don't know why, but I have less colors today than I normally do, but whatever. External regulators make sure to find out whether or not you need to go faster or slower. Basically, they look at the outside of your body, so this determines speed of growth. And this idea is this: while your body is just chilling, so like my hand right now is just fine. It is external regulators are telling my hand to regenerate skin cells as I need. So cells die, and we all have dead skin and that kind of thing. So like cells are just dying constantly and rebuilding constantly while I'm just sitting here, right? So that's a normal pace. But if I were to cut my hand open, cyclins would immediately react and tell the body that we need to have a lot more healing going on in my cut hand. That's what forms scabs so quickly, like that and vitamin K, of course. But it causes scabs due to clotting and whatnot. So it prevents anything from entering as fast as it can. So the body is all about internal and external regulators for cell division. And the internal regulators decide whether or not the cell is ready. The external regulators tell whether or not we should go faster or slower in creating these things. And there are all sorts of issues with these regulators, and they do this and that. Cancer is actually a really good example. So if your external regulators are all kinds of messed up, what happens is with cancer cells is that it tells your external regulators, hey, you need to reproduce really, really quickly. So your cells just reproduce like crazy, but your internal regulators are not deactivated, but they act in a weird way. So the external regulators in cancer tell you, man, we need to create more cells really fast. So it starts to create cells. The internal regulators say like, okay, cool, so we're just going to make these cells, and we're just in too much of a rush. They're going too fast. They can't checkpoint them all. You just end up with a lot of really messed up broken cells or cancer cells. And that is why you see them just spread like crazy and get bigger and all that kind of jazz. So this has a lot to do with how cancer works and all that. Alright, so let's go ahead and move on to page 11, which is a question. Just kidding. Let's move on to page 12. And they're going to talk about the different checkpoints and whatnot. We're going to go ahead and talk about these different checkpoints. Now, this C thing, you're going to see it over here in, I'm going to start a new page. You're going to see it in this tutorial. C is, they call the whole cell cycle and these cyclins and whatnot. There's a post-test question about the C checkpoint. If you look it up anywhere else, like if you Google it and whatnot, nobody teaches a C checkpoint. It's just Plato as far as I know. So make sure to be really careful with any questions where you see the answer C in it. For the post test, I did post the correct answer for that question. So once you get to this test, I don't remember which post test it is. It's probably this one. It might be the next one. So it's either post test 3 or 4. Take a look at the study guide that's posted in the Dropbox, or ask somebody for the study guide, ask me, and we'll make sure to get you that study guide. It makes a point about this C phase that kind of exists, doesn't really exist. Plato says it exists. Alright, so we'll talk about that later. But let's talk about the other phases, like G1. Alright, we're going to start with G1. I don't know, I'm going to use, what's this color? Let's give this color a try. G1. Okay, cool. So we're going to use G1 with this turquoisey, what's it called? Teal. Just kidding, this is teal. Okay, we're going to start with this. G1 is just a growth phase. Alright, they're going to build up new proteins. That's it. So this is the growth phase. It just builds stuff. Alright, and we've talked about this before, so I'm not going to stress it too bad. But this is where the cell decides, all sorts of different things, like what they can do and what they can't do, and etc., etc. So G1 phase is really all about growth. Nothing's going to happen until you get enough growth. So you have to be big enough. Must be big enough. Alright. It's kind of like a roller coaster ride. Railroad, what in God's name? Anyway, a roller coaster ride where the people at the front have to check you to make sure that you're tall enough in order to go on the ride, right? And sometimes they let people through that are a little too small, and that would be an error in this case. So that would be where people start dying and falling off roller coasters. That's how you end up with unusually small cells or useless cells and that kind of thing. Alright, so G1 is the growth period. And let's talk about S phase. S phase is going to be very, very similar to G1 in a way. They have a lot in common. This one's going to be called the synthesis phase. So while G1 was growth, this one's going to be synthesis. Alright, and this is just going to double the DNA. Alright, doubles DNA. That's its job. So these two steps together are in charge of making sure your cell has all the necessary major parts. So they have to have enough growth, so they have to be big enough, and they have to have enough DNA. Because if you don't have enough DNA, you're going to end up with two half humans, which doesn't really make sense. It's just not going to work like that. Alright, so make sure to know these two as the starters for everything. They're kind of the leading idea. Okay? Let's see. Let me go ahead and check. I'm going to go ahead and hit this G2 button. Let's see what they have to say here. Cell enters the next phase of the cell cycle, G2 phase. At this stage, a cell which has made a copy of the DNA. Oh, okay. So in G2, you've already made a copy of your DNA. So at this point, all there's left is to make sure that you have everything else. So it kind of packages all this stuff together. So G2 is a much more specific version of G1, which we talked about in the last one. So here's G2. We'll use this blue thing. So G2 is what was I talking about? G2 is it's still G1. It's growth, but it's more specific growth. It's more specific. And like I said, we talked about this in the other tutorial, so I'm not going into massive detail about this. I'm just looking through the mastery test to figure out what exactly it is that you need. So that's G2, and then let's talk about the M phase. And the M phase is real simple. This is just a splitting. What color? Green? Yeah, green is what I have to use. So, sorry about that. M phase is that mitotic phase. This is that splitting phase. So again, splitting phase. You guys know all of this if you've seen the other tutorials. It's exactly the same. I'm just going to go through the mastery test. I'm going to talk about a few things that might throw you off that weren't on the last mastery test. Like really, really specific things, right? So this is a general overview. There's that part. There's this part. Alright, main thing I want you to take away from this is cyclins are timing proteins, so they determine like how fast you split, when you split, etc., etc. Internal versus external. External determines how fast you split. Internal determines whether or not you split at all. Okay, so you have to go through both of these. You have to, you have to get through this stage to get through this stage. So I gave you the example of cancer, screwing all this up. So cancer makes the internals like, everything's cool. We'll just go ahead and release it. External cancer says we should grow really fast. It's as if you're hurt, kind of like if you have a cut in your skin or something like that. All right, so let's see. We talked about the different phases. Oh, they actually have a cancer question in the mastery test. You're fine. You may or may not get it, but that'll be okay. Internal and external regulators. We talked about these. These are your proteins. These are your cyclins. Okay. Just keep in mind, anything that causes cancer, I don't know why this is in your mastery test. It's probably somewhere in your tutorial, but the word for anything that causes cancer, so, they say smoking leads to lung cancer, too much skin, too much sun exposure to your skin causes skin cancer, all the other kinds of cancers. Anything that causes cancer is called a carcinogen, okay? So like tanning beds, they consider carcinogen, C-A-R, I guess I should write it down. C-A-R-C, N-O-G-E-N. Carcinogens, they cause cancer. Cause cancer. And this can be anything, this could be alcohol if that causes cancer. Pretty much anything that you can figure out causes cancer is called a carcinogen. Alright, on this mastery test, what else? Oh, keep in mind, during that healing process, so like let's say I got a cut, right? The external regulators would at first make my regeneration really, really fast. So just to get that scab on my hand, that scab part is going to be really, really fast. But scabs stay there for a while. You can get a scab. Your skin will heal up really, really fast. But the scab will stay there for a long, long, long time until you end up with like a scar. And that scar will stay there for pretty much ever. So cyclins at first speed all of your cell regeneration up like crazy, like really, really fast. But towards the end, they start to slow down. Okay, so just make that note for your mastery test. Cancer cells, they metastasize. That's what they do. So carcinogens cause cancer, which cause metastasis. Metastasis is the spread of cancer. So this is spread of cancer. Not all cancer cells spread and metastasize. Some of them are called benign, but that's okay. That's not on your mastery test. But benign cells really don't do a whole lot. They just get really big and they're hard and they're just kind of there in your body, so you should probably have that removed. They're not particularly dangerous, but they can get in the way of your body's regular functions. So it's best to get rid of them early on. What else? You know what, that's it, honestly. They talk about cyclins towards the end, but that should cover pretty much everything for your mastery test. You ought to do okay. I guess this video went on for 15 minutes after all. I expected it to only take like 5-10. I probably should have just started with the mastery test. But yeah, a lot of this was review, so, you know, feel free to look at the other tutorials if you like, but we've already covered all this before. Anyways, best of luck, and contact me if you need anything.